Hello and welcome, everybody. It's wonderful to have you with us today, and it's actually fantastic because this is our encore presentation. And before we get into the webinar today, I'll just mention that we're very much looking forward to being able to have this as a conversation after the webinar, and to be able to host some Q&A. And what we'll ask is that if you're able to press the Q&A button, or you can dial *6 to get in the queue to ask a question, then we would love to hear the question directly from you. As a backup, you can certainly put it in the Q&A, kind of type it in. But we would love to hear from you, and so we'd ask that you click on that. But one thing I will mention is that you may need to associate your call and phone number with your web login. So, for example, you can see on the screen here, this is just a number unassociated with a person. But when you dial in, you'll have a little pin there that you hit the # afterward, and then it associates it with your login. My understanding is that when you do that, then this button comes live for you, and then during the Q&A session, you'll be able to do that. So I hope everyone's able to do that, and we look forward to hearing from you. Our moderator for today is Dr. Nisarg Modi, and he'll be moderating both parts. Today's part is Rheology Essentials for Pharma Scientists, Part 1, which are the core principles and the terminology. And with that, let me also just say that I'd love to take a few minutes for those of you who don't already know Dr. Modi. He's the Director of Research and Development at Transdermal Research Farm Laboratories, or TRPL. He completed his Bachelor of Pharmacy from Sardar Patel University in India and received an MS and PhD from the Division of Pharmaceutical Science at the Brooklyn campus of Long Island University, or LIU. During his tenure at LIU, he worked on the development of semi-solid products and the evaluation of their internal structural properties using rheology. Dr. Modi has expertise in developing TDS for new chemical entities as well as for drug products approved through the 505(b)(2) pathway. Really, in addition to his remarkable innovation and deep expertise in transdermal drug product development, he also has a fascinating breadth of experience in other areas of the pharmaceutical sciences. In addition to the development of successful commercial products, his innovative work in transdermal drug delivery has resulted in numerous patents. And I'll say that Dr. Modi is also the secretary of the AAPS Topical and Transdermal community, and without the enormous amount of hard work and dedication to our community that he's provided, I can honestly say that we would probably never have been able to provide an entire year of fantastic monthly webinars that we've consistently brought to you. We are very fortunate to have Dr. Modi with us to moderate today's webinar, and I'll hope you'll join me in welcoming him. Dr. Modi?
Thank you, Dr. Rane, for a very nice introduction. Today we have our presenter, Neil Cunningham. Let me introduce Neil. Neil is a very highly experienced consultant in the application of rheology and viscosity testing techniques across medical device and consumer healthcare. Since starting in the field of rheology in 1995, he has successfully consulted for the world's leading manufacturer and has trained and advised thousands of scientific personnel in the principle and practical application of rheology and viscosity testing. Over that time, he has gained an intimate knowledge of the most popular rheometer and viscometer in the marketplace, as well as a unique breadth of understanding of the need of industry. Throughout his career, Neil has also worked with the instrument manufacturer, including TK Instrument and Brookfield, providing consulting, instrument designing, and webinars and training courses for them. Neil has a reputation for his accessible, enthusiastic approach to rheology and his ability to convey seemingly impenetrable concepts to even the most non-technical person. Currently, Neil is the CEO of the Center for Industrial Rheology, which he founded in 2012, and which has grown to be one of the world's most trusted and respected laboratories, providing research services, contract, sample analysis, training, and consulting services. We are very fortunate to have Neil with us to help us explore Rheology Essentials for Pharmaceutical Scientists, a very special two-part webinar. Please join me in welcoming Neil Cunningham. Neil?
Thank you very much, Dr. Modi. I really appreciate that introduction there. Good morning, good afternoon, everybody. It's really nice to be here. I really enjoyed presenting this the first time round, and I'm hoping it will be equally interesting the second time round. And then I'm also looking forward to tomorrow's session, where we're dealing with the more practical aspects of rheology and viscosity testing. So, Dr. Modi, can I just confirm that my screen can be seen, my presentation can be seen?
Yes, Neil, we can see it well.
Fantastic, wonderful, thank you very much. Okay then. Okay, so rheology is it can be a difficult subject for many people. I remember way back in the mid-90s when I was learning rheology. I was plowing my way through textbooks with lots of heavy maths and formula and equations, and it never really struck me how best to sort of convey this. I was a sales guy back in those days, actually, and I was selling rheometers, and in order to be able to sell a rheometer, I had to be able to understand my customers' issues and understand their problems, but also to be able to convey these concepts in a way that made it real to people. And it was only when it finally clicked to me that really all a rheometer is doing is feeling a material, and it's doing that in a reproducible and quantitative, objective way. Then I started to get a real handle on how best to kind of talk about these concepts, really. And if you think about rheology with respect to pharmaceutical and healthcare products, then there are a whole bunch of ways in which rheology can impact on those products. Anything from delivery and handling and pouring, pumping, stability, sensory acceptability, those sort of areas, really. So, and what is really fortunate to us then is the fact that we're surrounded really by a whole bunch of materials in everyday life that we can use as learning opportunities to get to grips with rheology. When I run my practical training courses for companies, I find myself in the Walmart the night before, buying up a whole bunch of different products that we then lay out and present to a group the next day. And I usually lay them out, and I've usually got things like this jello and marshmallow and soap and creams and things like that. And I lay these out and then ask a few sort of challenging questions about these products, really. So these are the typical kind of things that we'll be looking at, really. First task I usually set the group is, could you separate these products into liquids and solids? So if you think about these products, could we separate those into liquids and solids? And it's very difficult to do that then, really, because on the left-hand side there, we usually get the marshmallows, and the jello goes into the solid category. On the right-hand side, things like the soap, the honey, the liquid soap. But then we end up with this category in the middle here that everybody wants to throw everything else into, really, semi-solids. And that tends to be a bit of a challenge, really, because description of semi-solids to some people could be different to what it is to others. So another task I often set is, we'll take these materials and then put them into a ranking where you've got most solid at one end and most like a liquid at the other end. And then typically we'll get something like you can see on the slide here, with something like the Vaseline. It feels hard, it doesn't flow, so it's usually put at the most solid end. And the most liquid end, then you'll get the tomato juice or a smoothie or something. But again, that can be difficult, because what we can see is that something like the hand soap, it flows like honey, but it also wobbles like jello. And the similar sort of thing you can see with the tomato juice. There you've got something that looks like a liquid, but in actual fact, you could stand a spoon up in it. And so this can present us with some difficulties in categorizing materials. And I'll just show you another example of that. What we can see here, on the left-hand side, you can see some chocolate milk. On the right-hand side, you see some pretty thick honey. Okay, so two very different products in terms of viscosity. But what we'll see then is that the chocolate milk on the left-hand side, you can actually get a spoon in there and you can get it to support the weight of that spoon. So you have a structure in here that's supporting the weight of that spoon, whereas the higher viscosity honey is not able to support the weight of the spoon. So there's something more to it than just looking at viscosity here. I've got another example here. On the left-hand side, we could see some mayonnaise. And what we can see is the mayonnaise, it bounces and wobbles like a solid. So it wobbles like jello, but we can make it flow. If we give it a push, we can make it flow like honey. Momentarily, it will flow like honey. On the right-hand side there, we can see some hand soap, and it flows like honey. But then we can make it bounce and wobble as well. So if you start asking the question, are these more solid or more liquid? What's going on here? We can see here that there are some complex behaviours that we really need to capture and understand here. So what I'd like to present today really is a simple, practical classification that we can use for describing these materials. And we could take suspensions and emulsions, and we can describe those as structured liquids. And you can think about structured liquids as being solids that can be made to flow. If we compare those to polymer solutions and surfactant solutions, a simple description of these, we can call them elastic liquids. It's not a perfect description, but it's a useful practical categorization. And elastic liquids can be thought of as liquids that can be made to bounce. Really. So what this does is it gives us a simple framework that can help us, guide us with our choice of rheological metrics that are likely to be most relevant for any product that sits into those groups. So suspensions and emulsions typically exhibit this structured liquid behavior where you have this soft solid structure, but then you can make this flow with the application of sufficient force or sufficient stress. And polymer solutions and surfactant solutions typically occupy this elastic liquid category, where they'll pour and they'll flow, but you could also get them to show that elastic bounce and wobble. The other thing that you see in elastic liquids, and it's a little outside of the scope of today, but you see a lot of stringiness and stretchiness and extendability. And that can be highly relevant for lubrication and coating and film forming, and can also be problematic in processing as well. But that's something that we could talk about at a later date. So, okay, so I'm going to focus on structured liquids because structured liquids tend to be most relevant to the world of topicals here. So what do we need to know then? What do we need to measure and quantify in order to accurately describe structured liquid? Well, what I've got here is a list of the key characteristics, key rheological characteristics that we want to capture. Okay, and we're going to be going through these one by one throughout the rest of the presentation here. Before I do that, I just want to cover just a super quick recap on some theory here. There are three metrics, three metrics that we need, or three definitions that we need to define here, really. Shear stress, shear strain, and strain rate, or shear rate. Okay. So whenever we either handle a material, or we assess a material, or a rheometer measures a material, it's controlling one of these and it's measuring another. Right? So shear stress is basically the force that's been applied to a material divided by its area. Okay, so we apply a force to the top area of this. Imagine that we've got some jello in this gap between these two surfaces. We're applying a shearing force, so a tangential force. That force is spread across that area, and that generates a shear stress within the material. If force is measured in newtons, area is measured in meters squared, then we get the unit of pascals, newtons per meter squared, or pascals for shear stress. If this is a solid, suppose this is a chunk of jello in here, we apply the stress to it, then we're going to get a certain strain value. So we're going to get a certain amount of deformation. Strain is how we quantify that deformation. Okay, and the strain is basically this displacement divided by the thickness of the actual amount of product that we have there. Because it's a distance divided by a distance, it's unitless, but we often turn that into a percentage here. Okay, if we don't have a solid in here, imagine we have a liquid instead, and we apply a stress to a liquid, then what we're going to see is we're going to get an onset of deformation, but that strain is just going to keep on increasing with time. So then we can define and quantify a strain rate or a shear rate here. And so shear rate or strain rate, they're one and the same. And this is basically, it's the rate of change of strain with time. Another way to think about shear rate is it's the velocity gradient. So it's the velocity divided by the thickness of that shear field there. And this can be useful when it comes to making decisions as to appropriate shear conditions in which to run a specific test. Okay, so we've got our quantities defined here, shear stress, shear strain, and strain rate. And then we just need to understand two other properties. Number one is modulus. So rheology is a study of the flow and deformation. Deformation relates to solids, and modulus is essentially the rigidity of a solid. So modulus is the ratio of how hard you push to how much deformation you get. So it's the stress divided by the shear strain. And this was defined by Robert Hooke, and this is where we get our modulus value. So shear stress is in pascals, shear strain is unitless, so modulus, G, if this is in shear, is also defined in pascals here. And we've got some examples down here of some moduli for some everyday materials here. Okay, so this is for solids, modulus, measure of rigidity or resistance to deformation of a solid. For a liquid, we have viscosity, measure of the resistance to flow. And so the resistance to flow is basically the ratio of the shear stress to the shear rate. So it's the ratio of how hard you push to how fast the material flows. And this is Isaac Newton's famous equation here. Now, Isaac Newton noticed that some materials exhibit a linear relationship between shear stress and shear rate. If you double how hard you push, you get twice the flow rate, twice the shear rate. So that means that the viscosity is just a constant value. It's just a constant value there. But fortunately for us rheologists, most of the world is made up of non-Newtonian liquids, really. So it gets a bit more interesting than this, really. So okay, so we defined our properties of solid and liquid, so the rigidity or the modulus and the viscosity. So what we can then do is we can go back to our palette of metrics that we're going to use for characterizing structured liquids and make a start on this. So we'll start off with viscosity. We look at viscosity across a range of relevant shear conditions. And this is really where we have to start to take a look at non-Newtonian flow. So what we can see in this little video I've got on the top left here is we've got a dollop of mayonnaise again, and next to it a dollop of syrup here. The mayonnaise is standing up of its own accord. It has a high viscosity as a result of all the structure that's present there. The syrup, on the other hand, flows out under its own weight. So under these conditions, it has a lower viscosity. But the situation reverses when you go to spread them. So the low viscosity syrup now has sufficient viscosity to drag the plate across the table. Whereas the higher viscosity mayonnaise all of a sudden loses that viscosity, and so you're unable to drag the plate with the spoon here. And this is a really nice illustration of the difference between a Newtonian liquid, the syrup, and a non-Newtonian liquid, the mayonnaise here. And if we look at the plot on the right-hand side, here we're looking at syrup and mayonnaise. We're running what we call a shear rate sweep here. So we're looking at viscosity across a range of different shear rates. And we can see at low shear, the mayonnaise has the high viscosity. And so it's and that's why we get the mayonnaise can hold itself in a pile like that, really. As soon as you apply some shear, then this all shears down to nothing. It's a very low viscosities here, really. So what we say then is that the mayonnaise is a non-Newtonian liquid, and more specifically, we say that mayonnaise is non-Newtonian shear thinning material. Okay, so what we saw in that plot there is that we're plotting viscosity versus shear rate on a log axis here, and we're getting a straight line here, okay? But if we extend that shear rate range over a suitably wide range, then at low shear rates, we're going to see a plateau appear, and equally at high shear rates, we're going to see another plateau appear. So we have this region of shear thinning behavior, and then we have the Newtonian plateaus either side of that. We call the low shear plateau the zero shear viscosity plateau, and the high shear, we call this the infinite shear viscosity plateau. Yeah. And I just want to show you in some real data. This is a really nice illustration of these plateaus here. So this is a Nivea lotion. We've measured this over a very wide range of shear rates here, and what we can see is that clearly we can see the zero shear viscosity plateau coming in at around about 10,000 Pascal seconds. Now, it's just useful to think about what is 10,000 Pascal seconds of viscosity. Well, the viscosity of honey comes in around about 10 Pascal seconds. So 10,000 Pascal seconds is 1,000 times thicker than honey when you're in this at-rest condition. And I think this is a great illustration of the fact that there's a whole load of stuff that goes on when products are close to an at-rest condition that we don't quite realize when we're using simple sort of spindle-type viscosity measurements and those sort of things. Okay, right. So once we've got the capability then of performing these viscosity profiles across ranges of shear, then we can start to perform some shear rate sweeps. And what you can see here are some typical shear rate sweeps that we would perform on a range of emulsions here. And you can see these are all displaying a degree of shear thinning behavior, a significant degree of shear thinning behavior, and they're all displaying a fairly wide range of viscosities within themselves as well. Okay. What's important to note here is that some of these plots cross over, and that's really important. When you consider if you take, say, a single point measurement with a simple viscometer, then you could see the issue that you could potentially be running into here, in that the behavior at the low shear end could be the reverse of the behavior at the high shear end. So it's important to realize that viscosity is a plot and not a dot here, and we need to capture this. What we're doing in this particular test is a fairly simple, fairly limited shear rate ramp, and this is typical of what we perform in my lab here. And then we can extend that if we want to understand some other things. So the typical way to extend this then would be to start to look at the very low shear behavior. And the way we perform that is we typically then move over to, instead of measuring viscosity across a range of stresses, we measure viscosity, across a range of shear rates, as we did previously. What we then do is we measure viscosity across a range of stresses, and we just apply some very gentle stresses and just probe that structure down at those very low stress conditions. And we can see here we've got, again, another range of emulsions here, and they're all showing pretty significant shear thinning when you apply sufficient stress to them. So what we're doing here is we're going through these yield points, so we're breaking up that structure. But we're capturing these zero shear viscosity plateaus here. And so this is really getting into some extremely low shear conditions. And in tomorrow's presentation, I'll illustrate a bit more about the extent of our ability to get down into these kind of effectively at-rest conditions. Okay, so this is great. This is a really useful way of capturing this zero shear viscosity. You may have heard of creep testing. Creep testing is another way of capturing that. A similar sort of test, but what we're doing here then, in a creep test, is we take a sample and we just apply a very small stress to the sample, and we allow it to move over a period of time. So we're choosing a stress that's way below the yield stress. We apply that stress, and then we just monitor what the sample does over a period of time. And then what we can see from this is that we get a lot of deformation in the first few minutes, but then eventually our plot reaches a straight line, a steady state viscous, what we call a viscous creeping flow. Now, we know the stress that we're applying. The shear rate is the gradient of this strain versus time plot here. So that then we can calculate out the viscosity from this, from the stress divided by the shear rate. And we know because we're applying an extremely low stress to this, we know that that stress then is going to be close to the zero shear viscosity measurement for the product. Okay, right. So so far we've only talked about viscosity, but there's a whole lot more to rheology, and then when we start to look at the palette of metrics and what else we need to characterize to understand our product, we could take a look at some structural characteristics and viscoelastic properties. To do this, we need to enlist the help of oscillatory testing methods. Oscillatory testing is basically applying a very small oscillating stress to our sample. We're nudging our sample with some very gentle stresses. And what we do is we look at the response of the material when we apply those stresses to the material. So we're applying, in this particular example, we're applying an oscillating stress. So this is a sinusoidal oscillating stress, clockwise to counterclockwise. Imagine if we had a chunk of jello between the plates here. When we apply that stress, when we're applying the maximum stress, we get the maximum strain. When we apply zero stress, we get zero strain. So the strain is in phase with the applied stress, as I say up here, like poking jelly or bending a ruler or something like that. If, on the other hand, we had water or honey or a syrup or some oil between these plates, then we're not going to get that in-phase behavior here. There's going to be a phase lag here, and there'll be a phase lag by a quarter of a cycle for a completely inelastic, or what we call a completely viscous material here. So that gives us this scale, 0 degrees phase lag for a perfect elastic solid, and 90 degrees phase lag for a perfect viscous or completely inelastic liquid here. Okay. The other thing we can get from this is if we're measuring jello, we may get some strains like this. If we measure cheese, we're going to get much smaller strains for the same applied stress. And the same case over here, if we're measuring water, we may get some large oscillations here, large deformations and strains. If we're measuring a heavy syrup, we're going to get much smaller deformations. So we can look at the stress amplitude and we could divide that by the strain amplitude, and that gives us a modulus value. We call it the complex modulus because we're ignoring whether this is in phase or out of phase. Okay, but then we take that phase difference into account, and with a knowledge of the complex modulus and the phase angle, we can then resolve out an in-phase component and an out-of-phase component, what we call the storage modulus and the loss modulus for the materials. Okay, so storage and loss moduli, it took me a long time to get my head around these when I was first getting into rheology. And I've got a kind of here a pretty sort of unscientific, but more kind of intuitive approach to thinking about this. Imagine you have a sponge here, you soak that in water. Then you've got a certain overall resistance there. We could think about that overall resistance as our complex modulus. Some of that resistance is going to be a result of the contribution of the rigidity of the sponge. Some of that is going to be a result of the contribution of the viscosity of the water. And so you can think about these as our G prime and G double prime, the storage and the loss moduli, storage and loss modulus contributions to that overall modulus here. Okay, and they tie up in this way. It's not a simple addition, it's a vector arrangement here. Okay, but it's a useful way of thinking about this, really. And you can think that if you've got a more rigid sponge or a higher viscosity liquid, then your overall modulus is going to be higher as well, the overall complex modulus. Okay, so a very useful oscillatory test that we run very often in my lab is oscillatory stress sweeps. And oscillatory stress sweeps, what you're basically doing is you're gently ramping up the stress you're applying to a material, and you're looking at what happens as that structure breaks. So below the yielding point, we can see here. So these are all outputs of a measurement on the same material here. And what we can see, if we look, let's say, for example, we start down at the bottom left here. Here we can see the phase angle as a function of stress. We're gently ramping up the stresses. We reach a point when the phase angle, which has been low, which indicates that we've got largely in-phase behaviour or a structure present, we're then getting to the situation where we're breaking up the structure there, and the phase angle rises. As a result of the phase angle rising, we see that the modulus decreases because we're losing the stiffening effects of that structure that was present here. We can also look at that as storage and loss modulus as a function of stress up here. And here we can see that storage modulus is higher than loss modulus. And then when we achieve sufficient stresses, they then drop down and they cross over and drop down. And this is a point where our phase angle goes through 45 degrees here. The other thing we could do is we could plot this as a function of strain as well. So this gives us an indication of effectively the brittleness of our sample. A product with a high yielding strain is something that can be deformed a long way before it breaks. Okay, on the next slide I've got some examples of some other materials. Again, some typical cosmetic and topical products here. Here we're seeing the modulus plotted as a function of stress, and we can see a range of moduli here. Remember, high modulus means you've got a rigid structure, stiff like hard cheese. Low modulus means we have a very soft, very deformable structure, so wobbly like jello here. If we look at the same data as with phase angle as a function of oscillation stress, we can see the yielding behavior across this range of products. So we can see here that this Clarins cream has very high yield stress. It can achieve very high stresses, maintain very high stresses, withstand very high stresses before it yields. Down at the lower end here, we've got quite a bit lower range of yield stresses for the Nivea, and that's a good thing. We want Nivea to be pourable. We want to be able to pour a lotion out of a bottle. So the yield stress is a key requirement. A low yield stress helps us with easy pouring of a product. Okay, another oscillation approach is to sweep the frequency. So then we apply a small constant stress or strain, and then we sweep the frequency. And then what we can see here in this particular plot, very typical example of what we would get if we would measure something like this liquid soap here. So it flows like honey, but it wobbles like jello. Okay, if we think about that in terms of frequency, at low frequencies, the loss modulus dominates over the storage modulus, so it's more liquid than solid, flows more like honey, less like jello. At the higher frequencies, then the higher frequency oscillations, we see this crossover, and then we see this elastic dominance here. So, and this is a very useful test then for getting an understanding of the nature of the structure present in a particular product. What we can see here, for example, we have Zavirax and some generic equivalents from various places in the world here. And the Zavirax here, we can see we've got a crossover here. So we've gone from a viscous dominant condition, only lightly viscous dominant here, but viscous dominant at low frequencies, and then a crossover here, an elastic dominant condition here. If we look at all of these generics, we can see that the elasticity dominates over the viscous behavior across that whole range of frequencies. So that can give you some insights into the nature of the structure present here. So what I've done here is I've plotted just the phase angle, and that really shows clearly the difference between the Zavirax and these various generic formulations here. Then finally, something else we could do is we can sweep the sweep the temperature, and we could use this, what we call thermo-rheological profiling, to understand products that undergo melts. So things like white soft paraffin and waxes and those type of materials here. And here you can see temperature sweeps performed on a range of wax-based products, and on the left-hand side, we can see how modulus decreases as we get the melting occurring. On the right-hand side, we see the phase angle, highly structured, low phase angle at low temperatures. We ramp up the temperature, we go through these changes here. In actual fact, these products were actually cooling. So they were cooling from a liquid condition down to a solid condition. And as they cooled, they went through a range of different behaviours with crystallizations and various solidification processes here. This is really useful. We've got some pretty complex looking data here, but in tomorrow's presentation, I'll show you a way that we got some really great differentiation and grouping and clustering of that data. Okay, finally, I just want to talk about Thixotropy. Thixotropy is all to do with the speed of breakdown and speed of recovery when we see changes in the applied shear. A highly thixotropic material is something that can be thought of as something that takes time to reach an equilibrium when you change the applied shear. So, and I'll talk about some examples of this in tomorrow's presentation. Typical way we look at thixotropy is, the traditional way to look at it is to perform what we call thixotropic loops, or loading and unloading plots here. And here we're ramping up the shear rate up and then down again, and we see this product, the red line here, this product is showing a high degree of thixotropy. We get an increase in stress. This peak denotes what we call a static yield point, then a further increase here, and then a drop down. And here we see a lot of hysteresis here that we don't see in the product, is only showing little thixotropy here. The other way to think about this, or the other way to measure this, is to use a three-step test. And a three-step test, we're basically applying a low shear followed by a high shear and then followed by a low shear again. And what we can see here then is that a highly thixotropic product shows a long, slow breakdown when we apply the high shear, and a long, slow recovery following the cessation of shear. Whereas a less thixotropic product gets to those equilibrium viscosities much quicker. Okay then. Okay, it's been a bit of a whistle-stop tour of some key rheological concepts here. The key point here to understand is that we can get some great insights into the behaviour of our materials. For topical products, it's really useful to think about this structured liquid model. So a structured liquid being a solid that can be made to flow. And then when we're characterizing structured liquids, then we can start to think about, well, we're interested in the rigidity of the structure and the strength of that structure when it's present. We're interested in the viscosity of the material when you've broken that structure. And then we're interested in the rate of recovery back again to that initial structured condition when you cease applying any shear to it. Okay, so that's all for today. Short and sweet. Hope it's been useful and interesting for you. And Dr. Modi, I'd be very happy to answer any questions.